This is Sarnam Singh. We are going to discuss about the possibilities of using hyperspectral satellite data for forestry purpose. The area which I am going to discuss is basically the Doon Valley, where we have very different kind of forest, or you can say vegetation types, and they have various number of species which are dominating in different ecosystems. Before we go to the details of this thing, you have already got some idea about what is hyperspectral data is all about. What I am going to focus is how are we going to use this data, let us say for species typing we call it. So mapping of species in a forest ecosystem, that will be the main focus of my discussion. So let us try to understand what is a species. Species, that means normally we call it, let's say, mango or teak or sal. This is a vernacular name. Each plant has a botanical name, and botanical name normally we call it as species. And species, we can say, is a population of organism, that means a particular group of plants from which they can reproduce, that means they can breed together plants and then produce the what you call the or rear the fertile progenies. They should not be infertile progenies. So that is called a species. And we all know from the remote sensing that each species has its own characteristics. Let us say human beings are different from apes, or so let us say mango is different from lychee, or neem is different from, let us say, from any other plant. That means each tree species has its own characteristic. So let us try to understand. We have also been taught in remote sensing that the spectral signature of each of these plant organisms or animals is unique. So across a electromagnetic spectrum, if you look at it and try to study, you will find there are certain differences in the spectral signature of different species. But let me also remind you that even though each species is unique, but there could be lot of resemblance or lot of similarities between very close species, and therefore the confusion comes. And that confusion we can overcome if you talk about more data sets. That is why the hyperspectral remote sensing is all about. I just give you an idea about how the species are described. They can be based on the number of leaves. So let us say number of leaves could be 3 or 5, like in one pinus species there are 5, another pinus species there are 3 or 7, but morphologically they look like needle-like, but number is different. But from remote sensing we cannot count the number of leaves, so that is the limitation of it. Similarly, like color of the sepals, color of the petals, or even number of petals, number of sepals, calyx, these are the characteristics of the plants. Or even let us say even the surface of the leaf from where the reflectance comes, it may have hairs, it is different plants. It may be glistening, that means shining, it is different. It could be smooth, simple, it is different species. Or it could be very texture, could be very rough, it could be different species. So you can expect different type of the reflectance from different kinds of the leaf surfaces. The species can also be based on the leaf characteristics, such as leaf can be very thin, the reflectance will be different. Leaf can be leathery. Leathery means very thick, and they are not so flexible. The reflectance will be thick. It will be different from that. So these are different characteristics based on which the species is formed. And remote sensing doesn't give you all this information about the either anatomical character or morphological characters. What it gives you basically is the reflectance characteristics. So therefore we need to study the morphological characters, reflectance, and then try to correlate how best we can do it. Hyper means basically, I will not repeat much here, but hyperspectral basically means there are a large number of spectral bands. Basically this gives an opportunity to have narrow bands, and narrow bands gives you again an opportunity to differentiate between the very minor subtle differences between the different species. So the spectral signature which gets averaged over an area of a pixel, let us say, and if the spectral width is very long or very wide, then again you have the problem of very too much of averaging. So several species could be mixed up and you get an integrated value reflectance or DN or radiance, whatever you call it, and that's how it becomes more difficult. Let me also tell you this hyperspectral data initially was more applied to mineral detection and all that, but now it is picking up and we are also using for vegetation characterization. And another thing we should understand, the leaf direction, because leaf direction is a very important aspect of reflectance which we get to the sensor, and they are varied. And the age of the leaves is different, direction of the leaves is different, orientation of the leaves are different, size of the leaf in the same plants are different. That means you get a very complex nature of reflectance which has to be understood. Let us try to how it can be used. The satellite data, let us say hyperspectral data, can be used for let us say vegetation stress monitoring. It can be a drought or disease or insect, because any impact on the chlorophyll concentration or the palisade tissue or the mesophyll tissue or the leaf which is present there, if it is affected by any kind of the outside pathogen, it will have a negative impact. So we get a different kind of spectral response. Species identification, which I am going to talk about more here. Then we can also use this data for photosynthetic activity, because the plants have chlorophyll, and chlorophyll basically helps in the absorbing CO2, atmospheric CO2, and produces biomass, or let us say glucose in layman's language. And we can do a lot of work related to the productivity using this kind of data. And the very important application in forestry is the lignin assessment or the foliar nitrogen estimation of different species, because the presence of lignin in the system, or say what we call the biogeochemical cycle, basically determines the rate of biogeochemical mineralization. So if lignin is more, it has a different impact. So you can say it basically governs the whole biochemistry of the plant, which is decomposing or maybe lying on the bottom of the tree. It can also talk about the moisture stress, nutrient stress. We can also talk about the growth models and try to produce a yield prediction model. We can also have the assessment of the toxic substances in the soils and the leaves. And because of all these things, we have a very, because of the sensitivity of this thing, it has to be slightly analyzed in different way as the multispectral data is analyzed. Lot of studies are there. I will not be going into detail of it. People are using hyperspectral data for foliar biochemical concentration analysis in different ecosystems, because this biogeochemical cycle is very important in nutrient cycle in any forest ecosystem. And there are a lot of variables available to us. It can be a leaf area index, that's called LAI, leaf area index. That means what is the crown cover, number of leaves, let us say, per unit area in a system. And because it is directly related to the absorbed fraction of the photosynthetically active radiation, and then we can relate with several other things and try to do the productivity assessment or many other kind of studies. Therefore, the concentration of these components in the leaves, and then analysis of the chlorophylls or carotenoids present, or the cellulose or lignin, these are the very important aspects of hyperspectral data analysis. As I said earlier, lignification of the leaves inhibits the leaf decomposition. So if in any ecosystem, if I have to study it, we can assess how much is the nitrogen available in a canopy, how much is carbon available in the canopy, how much is lignin available, how much is phosphorus available in the leaves, because leaves normally fall and they decompose. So all this kind of analysis has been carried out using this hyperspectral data. And similarly, we can also work on the leaf water concentration or the cause of the reduction in the leaf reflectance. All these can be studied specifically. Therefore, the narrow bands which we get in the hyperspectral data, they are very useful, and you get very sharp changes. Those sharp changes in the spectral signature basically help us to find out the species or species typing we call it, and then you can use this for characterization of the entire vegetation. Let me also tell you why this is another important. If we can identify plants which are very rare, or which are the communities or plants of group of community which are very rare, then we can try to have a very specific management plan. So there is a spin-off of this kind of studies to have the application for forest conservation and biodiversity conservation also. Let me just give an idea why it is important. I think earlier speakers must have given you some idea about the spectral and the multispectral data and the hyperspectral data. Basically, the presence of large number of bands allows us to do lot of study. It can be related to biochemical, it can be stated to the land use change, can be impact of the anthropogenic activities, and can be identification of the different features, like it can be type of species, minerals or soils where it grows. And then we can also try to, in coastal regions they have done some work, then grain size in snow they have done work. So there is a lot of application of the this kind of data. Now one of the most important thing is the hyperspectral is the what you call the red edge actually, because this keeps shifting depending upon the moisture content. It will shift left or right, and because of the uniqueness of this spectral signature of vegetation, the uniqueness is that this is the only object you can say on the earth's surface where there is absorption in red band and there is a reflectance in the infrared band, near infrared band you can say. This inverse relationship, that means if R increases then NIR decreases, if R decreases then NIR increases. This unique relationship along with the moisture content, that means we have pigmentation zone up to let us say red, and then we have a cell structure zone, and then we have a water absorption bands. All these put together they lead to change or shift or change in the height or direction of the edge. So the spectral signature keeps on changing depending upon the conditions of vegetation. Therefore all these considered, we try to attempt how we can use this data for species level mapping, because my objective is that can we do this species level mapping using the this data, because when you take broader bits, let us say other any landscape data or HRS data, then the average and DN value has mix of several species. Even if you do the sub-pixel classification, you cannot identify the species. But when you have a very narrow bands, it gives you an opportunity to find out what kind of a species could be there. So this is a very important application of this thing. So what we thought actually to evaluate the applicability of hyperspectral data for the dominant species in the forest ecosystems of, let's say, we have worked in the Doon Valley, and the whole idea is that we should have a spectral library, because if we have the spectral library, we can use directly this library and anybody can use it if they are put on the web, and then they should be able to classify their forest or the area in that way. And then these maps could be used for all kinds of before I go into this, it is very important for us to understand what kind of climatic conditions are there, what kind of vegetation is occurring, because all these the type of species occurrence depends upon the climatic conditions. So let me show you, Doon Valley basically is in the just outer fringes of the southern part of the Himalayas. Rocks are quartzite, unconsolidated lime, there are many mixed ones. The altitude varies from 500 meter to 2200 meter. That means it has a representation of tropical to temperate forest types. So it has very hot in the tropics, and then to cold, that means you get snowfall in the let's say 2200 meter and around that. It has very high rainfall area. Temperature also varies from 0 to maybe 40 degrees Celsius. Humidity is also very high during the rainy season, and also experiences the snowfall. That means we can expect in this kind of area the plants which have very broad leaves, the plants or trees which have very narrow leaves, and the plants which are adapted to the very cold climatic conditions, or plants living in a very hot conditions. So we have a whole variety of ecosystems represented in Dehradun, and we can see the impact of basically the climatic conditions on the distribution and try to map these using the satellite data. Let us see what type of the forest we have in Dehradun Valley, for which we will be trying to map the dominant species. We have moist deciduous forest. That means these are the forests which shed leaves for only few days or maybe less than a month. And we have very dominant forests of the Shoria robusta, that's called the sal. Similarly, Termina alata, Syzygium, that's called the jamun, Acacia catechu, that's called the khair. Then we have the sal forest, which is almost pure in nature. And then we have subtropical forests where we have the cheel pine, Pinus roxburghi, and the other species. Then we have temperate broadleaf, and then we have temperate coniferous, that means narrow leaf plants. So we have tropical, subtropical, temperate broadleaf, that is the oaks, and then narrow leaves, very needle-like leaves. They plants like deodar, thuja, or in the Pinus wallichii and all that. And the top of that we get patches of the grasslands where we don't find trees, but there are temperate grasslands which are basically used as a grazing grounds for that. So considering this thing, this is the background of this thing. Let me just tell you what we have been doing here. So this is the, just talks about where the location is. This is Uttarakhand here. This is the map of Uttarakhand, and then these are the two strips of the data, which Hyperion data, which we have used for this present study. These are the images which we have selected. So you can see here, this is one image of this Rishikesh area, which has very inner Himalayas, that is the temperate forest, two tropical forests, and this is the river Ganges, that kind of thing. So we can try to analyze this data. So we have taken a part of it for analyzing the species typing. These are the basically instruments which we have used. We used a spectroradiometer, that's called analytical spectral device, for taking the spectral signature of different species in the field, so that we can have a spectral library of different species generated. Then we used a very good GPS, because we should know, because you don't get single species plants growing at one place, except for few like teak and sal, but in other places you will have several species. So an error of 5 to 10 meter may result into a different species kind of combination. And then we used other CHN analyzer for analyzing the carbon content of the leaves, nitrogen content, then hydrogen and sulfur, because suppose I have to do the lignin analysis or carbon analysis of the leaf or the foliar carbon, we need to do all this kind of, and then try to extrapolate this information that cannot be labeled. Okay, so, and we have used the software there, because these are the normal NV software for analysis, and then you can use ADAS, ArcGIS, and that is the static, stat is the for static analysis source. So we have used the Hyperion EO and satellite data from NAR. These are the characteristics. Basically resolution is 30 meter, and we had around 242 bands out of this, and the range is around 350 to 2000 nanometer, which is a very good for forestry works. So for any satellite data, we need to do some of the pre-processing. The first thing is to have the data, and then normally we do the geometric and radiometric removal of the distortions, because they are very important, otherwise data will have a lot of errors. So the first thing is that there are a lot of lines which have been dropped. So first thing we do is remove the bad lines, that's the removal of the bad bands or de-stripping. Once that has been done, we then have to remove the atmospheric distortions. So we do the atmospheric correction. This can be done by using a flash algorithm in the NV. And then once we have rectified this thing, then we can do the geometric referencing, because in this case we have used the Landsat ETM data, which is already georeferenced, and since this also is of same spatial resolution, so it's always better to use this thing. Once we have done that, and then we go into the minimum noise fraction analysis, because this is again a kind of Fourier transformation, which forward and inverse transformation is done, so that all the redundancy is removed and we get the data which has least noise. And then we got now the field observations. As I said earlier, we have gone to the ground and we have taken the ground spectra, and once we have the spectra, then we have the GCP locations of that, and try to correlate and using the spectral angle mapper algorithm. So anyway, you can either we can take these values from the satellite data itself, that means pixel, or you can use the ASD values also. And once we have done this, then we try to classify it and do the validation and of course do the accuracy assessment of that. So these are the bands which we have removed. I will not read out because that is not of very this thing. So these are the number of bands which have been removed. They are non-calibrated, or they are overlapping, or they are bad in some way or other. So you can say after all, then we are left with around 158 bands which are used for processing. So first thing is the radiometric correction. So they are because of the sensor failure or atmospheric interference or reflectance. We do the reflectance to radiance conversion and then do the illumination correction, and then afterwards the geometric correction. So radiometric correction, first is the removal of the bad bands, and the column removal, that is basically simple. You can do in any either Mortron or Atrium or the Econ, any of these you can use, and then we can use the flash to remove the atmospheric distortion in that. You can see this is a very bad data, totally black. So this type of data has to be removed. This has negative values, so we should remove all these bands. If you look at this data, which is 94th band, let us say, there's a line, vertical line. You can see this is a line which has been column which has been dropped, and if you could zoom it just a little bit, you can see here this it is highlighted here. So you have 1 or 2 here, and then once you remove it, then this line is removed, and then after de-stripping, and then you have the average value of the neighboring pixels put here, and then you can use for processing. And you can see more clearly here, this is the line which is column which is dropped here. You can see the black line here, and after processing you can see the black line is removed. This is important if you want to keep the quality of your end product correct, because these black lines represent nothing, they have to be removed. Once we have that correction done, then we can take the landside ETM data or any other data which is available to you, or if you have very good GPS locations, if you know exactly where you have taken, maybe you can use that one. The whole idea is that we should have the data which is you have Hyperion data, you need to geometrically correct it. So that's how after the correction data becomes like this, little orientation changes. After that we do use the flash. These are the different parameters which are used for some of the important parameters. These are, you get most of these parameters from the header file, but some of the things you have to do, like CO2 concentration, you have to define. This is user defined. Normally it can be 385 ppm or 390 ppm, depends upon the area to area. Like Dune Valley is relatively clean, so we have taken 390 ppm. So most of the other parameters you can take from the header file. This is just a screen capture of the flash, how it is run. So you can see the different parameters which have been put here, and then you just run it and you get an output out of it. You can see here before and after the spectral signature of, let us say this is a profile of vegetation here. You can see a lot of distortions in the visible bands here, and not only in visible band, even this infrared bands also you have a lot of distortions. So after the flash, like this is the image after the flash, you can see even the contrast of the image has changed, and then you can see the spectra has also now has a normal trend. See, earlier this was showing very high, and this was showing also low. It was reversed. Now it is properly adjusted. So this is very important to normalize the data using flash, corrected properly. Once we have done that, then we are ready for the processing of the data. So there are many methods. The basic idea is that we would like to have the pure pixels identified. How do we do that? We have the GPS location of that particular plant. I have the spectral signature of that plant, so I know that this location, this species occurs, and then I can go ahead with the in-member selection and all that. So this, you can say, first step is the MNF, that is minimum noise fraction transformation. Then go to the pixel purity index, and then you can use any other, like we have used n-dimensional visualizer, that is a spectral angle mapper here, so that you can try to classify the image. What basically MNF does it's basically a principal component analysis, which you have the eigenvalues. So basically all your noise or redundancy is gone here, and then you get back again the same image by reverse. The pixel purity index is most important, because the species classification will depend upon how best you have been able to find out a pure pixel of that species. Now if you look at the 30 by 30 meter spatial resolution of this Hyperion data, you may not have many species or many plants with 30 meter crown. Therefore, in 30 meter by 30 meter, you will have lot of trees, maybe 10, 20 or 30 trees growing, and therefore it's really difficult. So you need to have go to sub-pixel level so that you can find out a pure, relatively pure index, pure pixels, so that you can try to have the collection of the. You can see here, this is the PC1, which has a lot of information. This is the PC2 and this is PC3. The information content decreases. This is a normal, we study in the multispectral analysis also. So this is basically reduces the dimensionality of the data set, and then we can go for n-dimensional visualization. This is very important because we need to analyze using different band combinations, and then you have different band combinations means you will have the cloud's shape changing as per the bands which you put in x-axis and the y-axis, and this is very important. And then you have to, we need to probably have those bands which, or those areas which represent that species. So selection of that, it's called feature extraction, basically selection of that band which helps in identifying a particular species is the most important of this thing. And then of course we go ahead, we have the spectral angle mapper here. This is very critical again. If I give the angle, that means with reference to the spectra, a very broad angle, then there is every chance that you have you are including the reflectance or the spectra of other species into your area. So that you may have a good classified map, but you may have a lot of errors in that. Therefore, this SAM has to be as narrow as possible so that we are able to map the species, because our objective is to map the species. We are not trying to map the whole forest because it may not be necessary. So it is very important, and this is an iterative process. We have to keep doing, maybe we do with 10% or 90% like that, and then the angle which you give 9, 10, 15, 20 or, and try to have the best result possible. Let me just give an example of this. Is here you can see the fine line, if this is there, then you have very good sal forest. All this red tone you get it here. This is a plantation of a teak, which is a little dark green gray color you can get here. This light pink in color you can see here all along. This is grassland, basically a small grass, and then you can see a dark patch here which is ipomia, we call it. This grows everywhere where there is a little bit of water, and it can. And then there are mixed plantations like mixed moist deciduous forest. Then we have different species growing here. These are plantation of the kanju, their khair, like that. You will have a lot of. Similarly, we have teak plantation also along the roads as well as along the block plantation. You can see here in more detail, and then you can identify even single tree also. So this is how we have collected the ground truth. This is of course not the Hyperion data. This we have used. This is a Iconos data where we have used to collect the ground truth. Basic idea is that we should go to that location and get the best spectra possible. So you can see here different features here. Like this is a banyan tree here, Ficus religiosa, and this tree we can identify here, but we tried to map it. We found it rather difficult because of the mixing with the neighboring species, because there is a road from 3 directions and there is a gate here from which goes to the office. So it is very difficult to map such kind of objects. Let me just tell you about the ASD. This is a spectral device. This is a little bulky one. This is an old example. Nowadays you get a very handy one which is very useful, and you can collect the data. You can see here it has a reference here with white plate. So first we do this normalization, and then we the gun is pointed on the different leaves. Like this is a Bahonia. This is for another species here in the ground. So different species we can collect this spectra of this thing. Reference spectra has to be as close to as 1, but when we really work in the field, we find the illumination conditions keep on changing, and therefore this keeps on moving, and particularly in bright sunshine it becomes very difficult to. But first thing is that we must obtain this a stable line, then only we will be able to get a very good reference point for further classification. So this is a very important aspect of it. The gun which is here should be about 4 to 5 inches away from the leaf, and it should be vertical. You should not keep it oblique, because if you keep oblique then you don't get the true spectra. It has to be vertical, focusing on that. I just give you some of the examples now. This is a Tectona grandis teak. You can see this is the tall trees of it. This is the young leaf of teak. You can see this is a young leaf which has a lot of hairs on it, and then you can see this is the kind of spectra is generated here. So this you can see here, these are some of the kinks which will be very useful for identifying this species. Like similarly, there are bamboo, like this is Bambusa rhodonacea. This is another species of bamboo, they call Dendrocalamus strictus actually. This is also bamboo species, but spectral signatures are slightly different. We will see when we overlay all these things. This is Mangifera indica, the common mango so this is spectral signature of this trend is here slightly different. And this is Pinus roxburghii. This is on towards the higher subtropical region, which has a very narrow leaf, like needle-like leaves, and the spectra is different here again. Lantana is a very common plant. Lot of work now is being done in most of the countries to map the alien species, the exotic species which are, they are growing from, they are coming from other countries and growing and occupying localities or forest areas in other countries. Like India has a lot of such species. Lantana caramera is one of them. We have Parthenium is another one. We have what you call the Epetorium is one species. So there are certain species which have occupied vast areas, but they don't belong to our country. So people are doing a lot of research using Hyperion data or hyperspectral data to map the extent and distribution of such species. This is Moraya koenigai. This is basically curry patta, which we use in our dishes. This is the spectral signature of this is an associate of sal forest, which is significant. Now, if you overlay all these and one, you can now find out the difference between the spectral response of different species. Okay. Now you can see here the angle here is changing, and you can see in infrared region this zone is very important for species identification. Similarly, we can see here this zone is very important. Here you have some species are very distinguishable, like this one dry grass is very characteristic here, but then other species you find a lot of overlapping is there. But the emphasis is that we are able to clearly demarcate or identify these different species which are occurring. So you can see here we have the Thusa sinensis, we have, let us say, Anogysus, we have Eugenia eugensis, Buchanania. These are the species which are found in the tropics of the Doon Valley, that is in the sal forest and surround adjacent areas. This is just tell you what are the different bands which are useful for this species identification. So you can have a list of the bands. This is little lengthy process. So let us say you have Soria robusta, or this is Chai, Thea chinensis, Cuscuta reflecta, Anogysus. These are different species in the forest ecosystem, and these are the points which are very important. These are the spectral range. These are the locations which are very useful for identifying this species. This is one of the Herculean tasks one must do, so that we know which of these bands out of 150 or 158 we need to use for classifying these species. Let's say this is a result of spectral unmixing, where we have different objects classified. So let us say this is the green, all that you can see, this is the grass, then you can see the tea gardens, then you can see the sal forest, this is only the sal forest, then we have the riverbed classification, and then we can also orchards, we can identify the orchards which are in Dehradun. We have the tea gardens here, and then we try to correlate, try to find out what is the impact of atmospheric correction, because if we don't do the atmospheric correction, then what kind of misleading results we can have. So this gives them just comparison of, so on the left side is the before correction. If I don't do any flash correction, then I get this kind of, and you can see lot of mismatch here, particularly in the this area, the tea gardens, they are all wrongly classified and something else is being classified. So of course sal forest is broadly classified because they are very uniform, but any feature which is very scattered, they are not properly classified. You can see here again the different objects, like this is for the tea garden, the shapes, this is the FCC, and this is FCC, this is from if classified from uncorrected data, this is after the correction. So you have the different kinds of the shapes, basically indicating different area estimation if you are trying to do this kind of mapping. This is for barren land. Again, you can imagine here what kind of data is here. So this is the correct, and this one after flash, it is very close to the original or the real one. These are the spectral profiles of the little temperate region. The earlier one I showed you, that was for the tropical region. Now here you have for the temperate region, that we have the different kind of species here. So we have, let us say, pinus, this, we have a Cryptomeria, we have a deodar, we call it, then oaks, we have quercus, and then there are other shrub species like viburnum is there, trema is there, pogostemon is a very, Thuja orientalis is very common plants in the Mussoorie hills. So let us see, this again region is very important for mapping all these species. I talked about the different angles, because when you talk about the classification using spectral angle mapper, then the angle is very important, and you may have to do lot of iterations before you arrive at the very appropriate spectral angle. So you can see here for oak we have quercus, we have the quercus leucotrichophorus around 40.4, roxbury 0.2, like that. For different species we have the different kind of it all depends. If a species is uniform, large area, then you can do it very easily, like agriculture, there's no doubt. Similarly, like a sal forest, no problem. But suppose you have a lot of mixing, then of course the spectral angle has to be iterated several times. You can see here the different types of the spectral signatures of different areas. They are representing the before and after correction for, let us say, teak, sal, sesam, khair, jamun and kanju. So there are different species, different kind of spectra before correction and after correction. It emphasizes basically the importance of the atmospheric distortion removal. These are again the area, you can see here, these are the for the tropical area, that is around the Rishikesh area, where we have again different kinds of forests. So you can see here the this region is slightly different, so you can see the absorption in the red band is not that good, so you have more reflectance here, and because of that there is a less reflectance in the infrared band. But these kings, like this king, like this king, these are the kings which are very useful for the mapping of the species. Okay, this is the same thing which is zoomed view of, so that you can understand, like this is teak, dalbergia, sesam, that is Soria robusta, that is the sal, jamun, that is syzygium, acacia, that is khair, holoptilia, that is kanju. So we are able to differentiate all these tree species in that area. This is a classified map. After having processed all that, you can now see here the results indicates a lot of good mapping of different species. Even in the sal forest, if you look at in the sal forest, you can have you see sal, mixed sal 1, mixed sal 2, mixed sal 3. That means these classes again can be subdivided. Multispectral remote sensing also helps us to do up to a certain extent about the classification of the mixed sal forest or pure sal forest, but you have better delineation of these things. Similarly for even teak, so you can get within the teak forest also we are able to find different types of the classes, maybe because of the age or because of the density. Similarly, like we have Ipomia plantations, I told you, we have pure sal identified, we have pure teak, then we have, let us say, sesam, map sesam, then shrubby species, and then we have khair, that is Acacia catechu, then Holoptilia integrifolia, that's another tree, dominating tree species in this part of the country. Jamun is a very frequent. So the whole idea is that if you compare with the multispectral data, we are able to do a better classification of the species. Now the most important thing is this is very useful information for forest managers. Let us say if they want to see a particular species, they want to conserve or protect or give additional importance as far as, let us say, in terms of productivity or in terms of the harvesting, they have a better area estimates, they have a better accessibility map through, and they know where this species is growing. So this helps in better decision making as compared to the multispectral data, which has you can get, we have to do a little more of homework in that one. Now look at the accuracy assessment. We have given, this is for the Rishikesh site which we have done for different classes here. Let us say this is sal, this is a mixed sal 1, 2, 3 like that teak. You can see here in most cases it's a very good assessment actually, and many cases it is more than 100% also. It all depends upon the kind of GPS locations we have taken, the kind of species identification we have done, because we need to take in help of a good forester or good botanist or a good taxonomist to identify that species, because taking spectra is a very simple thing, but if we have to identify that species, all the remote sensing experts may not be good in identifying a species. We need to identify a species properly, and you may have to take a spectral signature of different plants of the same species within an area, and then you may have to do the averaging, so that you get all variability of the same species of different age, different locations averaged, and then you try to compare with the spectra. That's a very important aspect of it. I probably missed it. When we have collected the spectra using ESD, we have taken 6 to 10 observations of the same leaves and different leaves, and then we have done the averaging. So you get a mean spectral signature which represents the particular species. There could be some variations here and there, but when you do averaging, it becomes quite representative. Okay, this just to tell you what is the future of the hyperspectral data uses. The data which we are doing this kind of analysis, it starts from a point. Let us say it can be a very small fraction of a leaf. When we do the carbon analysis or lignin analysis or phosphorus or sulfur analysis, the leaf portion is only around 3 millimeter by 3 millimeter. The spectra which we collect also is approximately the same size, so there is no problem there. But when I want to extrapolate this information, that is from a part of a leaf to a leaf, and then leaf to the whole canopy, for that we need to have special instruments. The picture which I am showing you here, this is taken using a instrument called HemiView. What it basically gives you, it gives you the total leaf area of a species. Let's say this is a sal forest, and what is the leaf area of this forest? And because leaf area, if I know the content per, let us say, per millimeter or per square meter of any content, maybe carbon or lignin or any, if I know that one, it is very easy for me to extrapolate this information. So this is a field of further research where we can, particularly in the forest areas. In agriculture systems, of course, it's a lot easier because you can always move around and take the spectra on the top of it. But here we can't go on in the forest ecosystem on top of the trees in the forest ecosystem. So we need to depend on such type of instruments where we can take the observations and try to do the modeling and try to convert the point information to a canopy information. Just to give an idea about what has been done, like this just gives an idea about the field measured nitrogen percentage, and then you have average, another satellite hyperspectral satellite data. So you can see you get a almost 1-to-1 match. It's a very good match for assessment. This only indicates that there is a high potential for hyperspectral data to be used for lignin assessment or carbon assessment, and we can correlate this with the ground objects, ground features or ground data with that of the taken from the hyperspectral data. This is an example, another example where we can have the whole canopy nitrogen based on the ground observations and then based on the hyperspectral data. So you have examples, people have tried to study the relationship, and they have shown very positive trend that hyperspectral data is going to be next data which will be used for species level characterization, then primary productivity estimation, and then let us say lignin assessment or carbon assessment, or even the carbon foliar carbon mapping of let us say an area, maybe a country. If you have data of the whole country, we can do the foliar carbon content mapping using this kind of a data. So this has a very high potential of. This is basically accuracy assessment of the this Masuri area where we have the temperate forest, and you can see here the user accuracy as well as producer accuracy in almost all cases around 100%, except few cases, let us say 66%, that is about settlement, because it gets mixed up with the soil and the sand, so you get a little less. But if you talk about the species like Thusa, Viburnum or Trema or Cedrus deodara or Cryptomeria or even Pinus wallichiana, these are the dominant forest species which form large patches, and we get nearly pure signature of these, and therefore they are easily to classify. So we can get a very reasonably good amount of classification accuracy which is acceptable within limits. With this I would like to say that the hyperspectral satellite data has a great potential, particularly in identifying the dominant tree species. Let me tell you why I am saying. Because any species which is co-dominant or which is found below the canopy, and you don't get a spectral signature, particularly in the dense forest. If it is an open forest, yes, you do get the spectral signature of those co-dominant and herbaceous or the shrubby species, but in dense forest we don't get this kind of. So we should start with the dominant species in the dense forest, and then of course in open forest we can have the other species also mapped. And the very important thing is, because in order to avoid the reinventing of the wheel, probably we need to have a national spectral library. We have been attempting to generate this kind of library in the part of Doon Valley. We have about 65 species, such kind of a spectral library generated. But again, this has to be done in different seasons, because the seasonal variability is very high, and phenology changes. Young leaves, old leaves, mature leaves, senescent leaf. So all these stages are there, and reflectance at each stage is different. Therefore, we need to select the same data set for the same for mapping this kind of. And then our study has shown that we have got a very good, fairly good accuracy, and the accuracy all depends upon the GPS location basically. If you have recorded your coordinates most appropriately, identified that tree most appropriately, and then collect the ASD spectra, then of course you can always expect to get a good accuracy. And before that, we need to have the geometric correction. Geometric correction has to be really excellent. It should be not more than half or a quarter of a pixel mismatch. If it is that much, then probably we may be standing in pixel A, and probably we are trying to collect the reference spectra and spectral pixel B. So this kind of course we can overcome these problems by taking a kernel and all that, but starting should be that we should try to collect the signature as best possible in the given conditions. Okay, with this I would like to thank you, and maybe I take up some questions from you. Well, there are a large number of questions. The first question is, explain about illumination corrections. Illumination is one of the problems in handling satellite data of hilly areas, because you have a lot of shadow effect. That is one, and one aspect, maybe northern aspect, is under shadow, and eastern aspect, southern aspect, or northwest aspect under lot of illumination. There are processes, like we can take some indices between the, like we do it for the multispectral data also. We can take up some indices to eliminate the shadow areas, or we can do the correction also. And there could be possibility to use normally they integrate the digital elevation model. So digital elevation model can be used for removing the illumination distortions, because if you have the proper DEM, then we can correlate, and then we can try to overcome this problem of illumination distortions. The second question is, how can we use hyperspectral data for forest fire analysis? That's a very good question, because we have in our country a lot of fires, particularly in the Himalayas, as well as in the central part of the India, where you have dry deciduous, or say, let us say, moist deciduous forest. Now, see, the whole thing is, if we, again the forest again depends on what kind of we have. If it is a ground fire, and if it has it cannot be sensed through the satellite sensor, then also it will be having some problem. But if it is a crown fire, we can certainly map it, because we will have no foliar, no what you call pigment there, no leaves, no reflectance. So they will be quite distinct from the neighboring vegetation which is not burned. So forest fire analysis has a great potential. Hyperspectral data can be used for that matter. Any data can be used, but probably you can do it more accurately. Of course, it will be better that we have the thermal band, but as far as forest fire, simple monitoring, mapping, damage assessment, that kind of, I think we can always use it. There's an immense possibility of it. The next question is how far it is possible to detect leaf area index using Hyperion data, since it has 30-meter resolution. Okay, let me tell you, leaf area index itself is a very complex process. It's basically area which is ground projected on the ground, the area of the leaves projected on the ground. That means we take a ratio of that, and the number of layers depend upon species to species. Like mango may have so many layers of leaves, so the leaf area index could be very large. Whereas there will be some trees, the canopy is very small, number of layers is very small, less, and then you have. So normally for leaf area index, we don't directly depend on that unless you try to correlate. So if you have, you can use either MODIS data, which already gives you the LAI map of that particular area, or you use the instrument which I have given you to, like HemiView, or there are other instruments like septometer. So if you can take the measurements, particularly in the forest area, because we can't go to the top of the canopy, so we have to take observations below the canopy, and that can be converted related to this Hyperion data, and you can convert that point data into a LAI based on the Hyperion data. So you can use it indirectly but not directly. Next question is, what if the angle difference in spectral angle mapper is so small, example they have given, less than 20 degree, how it can be differentiated? Yes, you see, as I told in the beginning, different species have different reflectance characteristics, but at the same time different species may have quite matching spectral signature, and if you have a very close matching spectral signature, it becomes really difficult. Therefore we need to find out a particular band. So let us say we have 150 and odd bands. Out of that band we need to do a very detailed inventory that which of these bands will be useful. Maybe in the infrared region, maybe in the middle infrared region, somewhere you will find some kind of a contrast which can be used. So that is a that takes time, but it is possible to do that. So normally we recommend we should take very narrow angle as less as possible, so that there is no spectral mixing and we get the species level typing or species level mapping as accurate as possible. The next question is, some of the species have similar spectral signature. In such case, how can we classify it using hyperspectral data? I think I have already answered the question, because the previous question is also related to that. Because if you have the spectral matching between two different species, we need to identify those bands. We need to go for some ratios. There are means to do that. It should be possible for us, because if we have a species and if we have a spectral signature or through ASD or by any means a good mean spectra, then you can, to large extent, you should be able to do that. Yeah. Okay. We have another question. How can the nitrogen fixing bacteria can be recognized through spectral radiometer, and so that the fertile and fallow lands can be differentiated by SAM for area calculation, okay, etc. Okay, let us see, nitrogen fixing bacteria normally are found in the roots, so they are underground. Now hyperspectral data, this basically spectral data gives you the surface information. So if nitrogen fixing bacteria is available on the surface, fine, you can get some spectra, but first thing, nodules are so small, okay, and then they are below the ground. Until unless you have taken them out and correlated with other soil parameters, that means maybe the nitrogen available outside the what you call the nodules, you can do to some certain extent. But again, you will have to have an indirect parameter, that will be your soil characteristic. It can be fertility level of the soil, or it can be nitrogen content in different of the soil, or the where you have nitrogen fixing bacteria or any leguminous species growing, and then pre and after you can go do that kind of assessment. Probably you can manage this kind of next question, how far hyperspectral data can be used in the conservation of forests in the verge of extinction like Western Ghats? Let me tell you, Western Ghats, as it is they are not going to be extinct so soon. We can talk about a species. There may be some species which are rare, threatened, endangered, or vulnerable to the anthropogenic activities of the human being. And if we can use hyperspectral data in conjunction with let us say niche modeling, or which are available, these programs are available on the net. If you can do this kind of modeling, we use the climatic data in that and use the satellite data, and then try to predict where they are available, and then we can go there and tell the forest department, look, that these are the ecosystems where you have such kind of species which could become extinct in the near future, and they could be conserved. But still we don't have that whole large area coverage of the Hyperion data or other hyperspectral data, but probably in the future, and very soon we will have the such type of data available for all the area, and then we should be able to do the species level mapping, and then species which are going to be threatened by human activities, we can prioritize them, and we can recommend for conservation. Next question is, can we estimate what is this? Diseased or infected species using hyperspectral data? Okay, let us try to understand what happens at the leaf level when there is any kind of infection. If we remember the leaf structure, it has the upper cuticle, then you have the epidermis, then you have the palisade tissue, and then you have the parenchyma, or the spongy air spaces are there, chlorenchyma is there. What I am trying to say, immediately the moment a infection takes place, the degradation of the chlorophyll takes place, the cells gets collapsed, the cells which are turgid, they get collapsed. What happens immediately? The reflectance characteristic, that means the interaction of EMR with the diseased leaves which have shriveled cells, which have infected cells, which do not have moisture, and if they don't have moisture, immediately you will have a reduction in infrared band and increase in the red band. So you can take up these some portions depending upon the species which you are looking for. Let us say it could be sal, or could be a even teak, like it has a lot of infection, particularly in Madhya Pradesh and all that. You have the which the leaves become like a net. So you can certainly use it. The only thing is you need to understand what happens at the leaf level. The moment leaf is infected, the whole physiological processes get modified. Leaves is no more turgid, and if this is no more turgid, means it will not have that percentage of reflectance. Let us say if it is 60% in infrared, it will come down to, let us say, 40% or 30%. That means, and then red will increase. Let us suppose it is 8%, will become 20 or maybe 15%. So this change in the trend of infrared and red bands will help you. That is one thing. If you are talking about the moisture loss, middle infrared band also will be very useful. That is the other side of two, we have the dips. These two dips also can be used for identifying those plants or those species which have been infected. So you have a very clear characteristic spectra on the visual FCC. Also you can make out that let's say if it is sal forest, you have very let's say very bright red color in young forest, or let's say little brown color, and if you have blackish brown color, that indicates there is some problem. We do not know what could be the problem. It could be because of the stress, it could be because of the infection, but certainly we can map that. Okay, thank you very much, listeners. Any question you can later on also you can post to me.